Welcome back, Year 12s, to another video. Alrighty. Alrighty, so today's class we're looking at creating human insulin through recombination. Okay, so welcome back, everybody. This is pretty much part 3 of the last 2 videos. So we're going to put everything we've learnt together into this one video to see what the process of creating human insulin is. Okay, so essentially what it is that you will edit a plasmid with a target gene. All right, you're going to try and get that plasmid into some sort of bacterium. So in generally most cases, Escherichia coli is the one. All right, and then we're going to look at how we can create the proteins, stick them together, and then we can make human insulin. Okay. At dot points as per usual. All right, so we're going to go through a process with different steps. All right, so creating the recombinant plasmid is the first step. So that's step 1. Alrighty, so plasmid vectors are prepared which contain the ampicillin resistance gene. Alrighty, so ampicillin resistance is shown in the green here. That's the antibiotic, ampicillin. So that's the resistance gene. Obviously, it has an origin of replication, as all plasmids will. And lastly, it has a reporter gene. So this is this blue one, the LAC-Z. Alright, and in this case, it's just going to have this one restriction site where a restriction endonuclease is going to cut right through. Okay, so that's step 1. Step 1 is essentially preparing your plasmids. All right, some sort of vector that we know has an antibiotic resistance gene and a restriction site found within the LAC-Z gene. All right, and that's going to come back later too. Awesome. Step 2, a plasmid vector is used. One for insulin subunit A, and obviously they're going to do one for subunit B as well, as you can see here. Okay, so what does that mean? That means that restriction endonucleases are used to cut that BAMH1 site, and we essentially insert this target gene, which is insulin A or insulin B. All right, this is going to be done in 2 separate processes, and eventually one of the last steps is they mix them together. Okay. DNA ligase is then used to connect the sugar phosphate backbone. All right, those phosphodiester bonds are reconnected via that. Obviously, it's a sticky end cut too, so the hydrogen bonds are also reconnected between the nitrogen bases. Okay, and there you go. You have your recombinant plasmid. All right, so that's the one with the orange target gene. All right, what's step 3? Creating transformed bacteria. So the plasmids are added to a solution of E. coli bacteria. All right, so they mix E. coli bacteria in some sort of liquid aqueous solution, all right, and you mix your recombinant plasmids. All right, then either heat shock or electroporation can be completed to increase the uptake of the plasmids into the bacteria. All right, so remembering heat shock and electroporation both have an effect on the plasma membrane of the E. coli bacteria, and hopefully they'll take up the recombinant plasmid. We know that some will take up regular plasmids that haven't become recombinant, and some won't take up any plasmids. All right, step 4. We're getting into the nuts and bolts of it now. So the bacteria cultures are spread and incubated onto agar plates. So you can see that here. They've put them onto agar plates that contain X-gal and antibiotic ampicillin. Alrighty, so the antibiotic ampicillin will kill off any plasmids that haven't taken up a plasmid, so non-transformed bacteria, all right, are going to be killed by the antibiotic ampicillin because they do not have the resistance there. Then we also have X-gal, all right, and that needs to get broken down. So colonies that form which are colorless or white, as we mentioned in the previous video, these ones here, any white colonies are the ones you're looking for. All right, are shown as transformed bacteria with the recombinant plasmid. Why is that the case? Because their LAC-Z gene has been broken, all right, because the target gene is inserted there. So they're going to make that insulin B subunit polypeptide, all right? They're going to make the protein, or at least half the protein we need. And therefore, the blue color isn't produced because the enzyme beta-galactosidase, which is made by this X-gal gene, which is made by the B-gal gene, is not breaking down this X-gal. Okay? Awesome. Recombinant plasmids will produce an insulin subunit with a beta-galactosidase tail as well, because remember, that's a gene. So the insulin subunit is essentially your target gene. It's inserted in there. But there is still the leftover blue parts, as you can see here and here. All right, that's going to make a pretty much a semi-beta-galactosidase tail that's left over. All right, so that's the blue part. Obviously, your target gene is the orange that we're looking for. Okay. This is formed from the half of the LAC-Z gene, which is transcribed and translated. All right, obviously simultaneously, because that's what occurs in prokaryotes, such as Escherichia coli. And yeah, so we're going to have this essentially insulin subunit B, which is the orange part, and then we also have a connected beta-galactosidase tail. So this is where it's a little bit different from the last two classes. This is where it's like the third part. All right, we're going to have to get rid of that beta-galactosidase tail. All right, transformed bacteria that contain the recombinant plasmids are then placed into conditions to exponentially reproduce. So we know they're going to reproduce exponentially, which means very, very quickly. All right, 2, 4, 6, 8, 16, 32, 64, etc. every 20 minutes roughly. All right, so after your 8 hours, you're going to have millions of them, and they're going to be producing millions of our protein chains that we have. Okay, so in this example here, the orange is subunit B, the red is subunit A, and then the blue on either side is that beta galactosidase. Okay, that tail. Before their membranes are broken down and the human insulin they have produced is isolated and purified. So pretty much you need to isolate off that beta-galactosidase tail. Okay, so you need to cut this out. So then you can get our insulin subunit A and insulin subunit B polypeptides coming together to form a quaternary structure polypeptide, of which is essentially our functional human insulin. Okay, so the two insulin chains have their beta-galactosidase tails removed. This is step 7, protein production and extraction, and they are mixed together. This allows the connecting of the disulfide bonds to form and create a functional human insulin. Okay, and that's what we can see down here through A and B chain. All right. Awesome. So this is the whole entire process in the 7 steps shown here. So again, if I just go through it quickly, we have our recombinant plasmid that a researcher's made in the lab. All right. Obviously, it starts with its origin of replication. That's not really important, but it is there. It has a resistance gene. In most cases, that's going to be ampicillin resistance gene, which is the antibiotic resistance gene. It has the LAC-Z gene, all right, which produces beta-galactosidase, and that's going to break down XGAL, all right. Also has a restriction site, BAMH1, which is in there, which means that's going to get cut at that restriction site. All right, obviously we have 2 processes going on at the same time. So obviously number 1 here, which is for subunit A, and number 1 here for subunit B. Okay, so we can just write A, we can just write B, boom, boom, cool. All right, so it's ligated to form this recombinant plasmid with your target gene. All right, so insulin B or insulin A. You then mix those recombinant plasmids with E. coli bacteria, and you perform heat shock. All right, so boom, heat it up, or electroporation, boom, electrocute it. Awesome. There you go. Some of the bacteria will take up the recombinant plasmids. Some will take up other plasmids which haven't taken up insulin B. All right, and then some won't take up plasmids at all. And you need to separate those. That's the whole point of essentially this 4th step. So bacteria transform with recombinant plasmids isolated from agar culture. All right, so we have our agar plate here. It has XGAL, which is a type of lactose that will be broken down. All right. Well, yeah, they can be broken down depending on if the recombinant plasmid is present or not in the bacteria. And we have obviously ampicillin antibiotic. All right, ampicillin will kill any of the bacteria which have just absorbed, just absorbed no plasmid. Okay, so they're non-transformed, but we still have to separate the transformed and the recombinant bacteria now. Okay, and that's where the Excal comes into play. So any bacterium that's taken up the recombinant plasmid, all right, that will lead to the breaking down of Excal and the production of the white there. I'm going to use black. The production of the white colony here, all right, because it's not breaking down that Excal. Whereas if it doesn't have the recombinant plasmid, it's going to be the blue colony. So it is breaking down Excal. All right, so you scrape some of that. You scrape some of those beautiful white colonies, and then you'll be able to get them in the next agar plate, and they're isolated. All right, from there, you essentially want to mix the bacteria together as well from both of the white colonies. We need to firstly extract the proteins. All right, so you take your protein out, and that's obviously our polypeptide protein that has both subunit B and a beta-galactosidase tail. All right, in this case it's subunit A and a beta-galactosidase tail. Once you've isolated them, we're on. Okay, we are absolutely on. Then we have our two proteins, which are then put together, subunit A and subunit B. All right, and we can mix them together and finally make our functional human insulin. All right. My suggestion, guys, would be watch this video multiple times. I know I'm really talking a lot, but hopefully it'll help it sink in. Okay, this is one of the harder concepts in biology, especially it really is one of the hardest concepts, okay, for the whole year that we're going to learn. So just keep that in mind. Keep doing what you're doing. You're doing a good job. Please like, comment, or subscribe if you think you got some value from this video. All right. Take care. Have a nice day.